സാധ്യം
Very good afternoon.
So the oral presentations will give you 10 minutes each. So you'll be timed. So Anil here is going to time you. If you exceed the 10 minutes, then you lose points. And then you can finish it before 10 minutes, that's fine. If you want, you can finish it in 5 minutes, that's fine. That's up to you. But you cannot exceed 10 minutes. And then there will be questions.
Okay, so we'll start off. I will announce the first 3 names so that the remaining 2 can also just be prepared just in case. So the first up we have Padma, who's talking about day-to-day drugs. Okay, so we then have Mr. C.H. Rajesh, PPT, next presentation. And then we have bioplastics by Navina. Okay, so yeah.
A very good afternoon to respected dignitaries, teachers and my dear fellow participants. I am K.H. Padma, currently doing my M.Pharmacy in Department of Pharmaceutical Analysis from AU College of Pharmaceutical Sciences. Today I am going to give an oral presentation on the topic Day-Grade Drugs, Detection Challenges and Advanced Analytical Screening Techniques. So these are the contents of my presentation. So firstly, what is a date grade? It is also known as a drug-facilitated sexual assault, where it is a type of forced sexual activity generally done by inducing the drugs to the person. This inducing of drugs is generally done through the drinks that we consume. This process is called as drink spiking. So coming to the day-grade drugs, these are the psychoactive chemical substances that incapacitate the individuals, making them vulnerable to the sexual assaults and making them incapable to fight back. So generally when the day-grade drugs are induced, the person becomes physically weak and also will not remember whatever is happening for themselves. Coming to the common traits of these drugs, these are generally odorless, colorless and tasteless. This makes them very easy to mix in the drinks that we consume without any notice. And they have a very rapid onset of action. They show their action within of administration into the body. They cause sedation, confusion, and loss of consciousness and amnesia. Amnesia means loss of memory. And these are the most commonly used state-derived drugs. The first one is GHM, that is the gamma hydroxybutyric acid. This is commonly known as the liquid ecstasia. Then the next one is rohypnol. This is commonly known as the Vlse. And then ketamine, commonly known as the cat Valium. And then scopolamine, commonly known as the devil's breath, and alcohols. Coming to the statistical data, it is found that for every 1 in 4 women are facing this sexual assault according to the National Intimate Partner and Sexual Violence Survey. And in all these cases, approximately 70% of the cases are involved using drugs and alcohols. And these cases are generally reported in most percentages in the nightlife events like the bars and clubs. And 60% of the reported cases are involving the use of sedative drugs. The best example is the recent incident on July 2025 where a student in the Kolkata school has drugged through the drink of a girl before sexually assaulting her. So all this statistical information and the events that are occurring has led to a need to develop a more sophisticated and advanced techniques like the portable and smart devices which can be used for the on-site detection of the drug presence in our drinks. So these are majorly of 4 categories. First one is portable strips. These are very similar to our litmus papers that we use for the pH detection. The wearable detection devices which are used as accessories and we can carry them easily everywhere we go. And the smart drinkwares, this includes cups and straws for detection of the drugs. And cosmetic detection tools where the commonly used cosmetic devices can also be used for detection of drugs. So the first one is the portable test strips. Generally we have a strip like this which is very similar to a visiting card. So it is very convenient for us to take it everywhere we go. The test is very simple. We'll just take the drop of our drink to be tested and place it in the spots A and B. And we'll wait for 30 seconds and we'll observe for the color change. If there is a color change, then it indicates that our drink is not safe for consumption. So here we can see the demonstration. Just we'll place the sample we have to be tested on the card and we can observe a color change. This indicates that the drink is adulterated with the drugs. And coming to the mechanism of action, generally on the cards, the reagents will be imbibed. And these reagents will react with the functional groups of the drugs, causing a color change. These are known as the colorimetric reactions. The commonly used reagents are 2,4-dinitrophenylhydrazines or ninhydrin derivatives for detection of ketamines, or we can also use pH-sensitive dyes. And this is another model. This is very similar to our pH papers. It has 2 regions, pink and yellow region. We'll apply the drug and we'll see for the color change. So here, as we can see, we'll take the strip and we'll apply the drink that we are consuming. As we can see, there is no color change observed, so the drink is safe to consume. The next category is the wearable detection devices. It is very similar to the strips, but it is more convenient for us to carry everywhere in the form of bands. It works on a similar principle. The next one is the ring detectors. This is not a marketed formulation, just the idea is patented by the University of Hawaii, where miniaturized chemical sensors are used for the detection of the drugs. So along with the colorimetric principle, the electrochemical biosensor principle is also used, where we can connect the sensors directly to the smartphones. safe for consumption, and if double lines are observed, then the drug is very safe for the consumption. Next comes the pocket NIR spectrometer. Here, when we take the sample and place it in the pocket NIR spectrometer instrument, it analyzes, and based on the absorption of the IR radiations, it gives us a unique spectral fingerprints for each drug. And the signal will directly be detected through Bluetooth by our phones. And our phones gives us a notification whether the drink is safe for consumption or not, like the warnings. So the next category is the cosmetics. In cosmetics, we have two types: lip gloss tubes and the nail paints. So coming to the future directions, we can integrate all these above seen portable devices with smart nanotechnologies or AI machine learning algorithms or lab-on-a-chip systems, and we can also use advanced spectroscopic approaches and integrate our instruments with the IoT and also nanotechnologies. Coming to the limitations of the portable devices, generally the most common and the most effective limitation of these drugs is they only can detect specific and few drugs. Like the most commonly used drugs like ketamine and GHB are detected most commonly. But some other sedatives like benzodiazepines, opioids and alkaloids and alcohols are not commonly detected by all the portable devices. And it has low sensitivity. These can be overcome by using nanomaterials like gold nanoparticles or quantum dots. So based upon all these approaches and limitations that are found out, we are working on a project proposal where we wanted to integrate a simple paper strip with the nanosensors and smart camera and AI apps, where we use the nanomaterials to overcome the drawback of limited drug detections. So the main objective of our project is firstly to overcome the limited drug detections. So we replaced it with the nanomaterials. These nanomaterials will help us to detect a broad range of drugs simultaneously at once. So the next aim is to create a safer environment for the women. This can be done by integrating our instruments with the IoT and AI instruments and smartphones. When we integrate the IoT applications, whenever a drug is detected in our drinks, the smartphones will directly send messages for our closed ones, ensuring the safety of the women in the society. So the only aim of our project is to build a world where science makes every drink safer and every woman stronger. So this is the overall overview of our project idea, where we take a paper strip and integrate nano coating materials in a multi-zone manner, so that when we apply the sample, they spread in these multiple zones and multiple drugs will be detected at the same time and are detected by the smartphones.
Can you make it faster? Your time's up.
I would like to conclude my topic by saying that the day-trade drugs are silent but yet a severe threat for the safety of the society. Even though effective detection techniques of day-trade drugs are facing many challenges, the techniques like portable devices from wearable sensors to app-based analysis are proving to be important tools for transforming the detection and also increasing the capacity of instant screening. These innovations are converting the lab-based techniques to the palm-based devices. The strongest tools against the drink spiking and the drug-facilitated sexual assaults always work together with the prevention, technology and detection. So I would like to conclude by saying that the future of drug detection technologies is moving towards a world where the safety is wearable, detection is instant and awareness is global. Thank you. I would like to thank everyone for giving me this opportunity, and I would like to thank my HOD for encouraging me to participate in this event, and thank you for your patient listening. Thank you.
Does anybody have any questions?
You have a prototype?
Sir, actually we are working on the project. We are in the initial stages. We only had an idea, sir. We need to convert it into an entrepreneurial project.
Anybody else? Any questions? Are there any patents involved?
No, sir.
You haven't filed any idea
No, sir.
patents?
We just have an idea in our hands. We have to convert it.
Okay, thank you.
Not yet tested. Whether it is tested or not? Whether it is tested or not?
No, sir.
Only the ideation stage.
Yes.
Any further questions? Okay, thank you.
Next up, we have CH Rajesh.
First of all, I would like to thank to giving the opportunity to present my AI-driven hot data protection framework. So as a team, we work as an three people, Paula, Dinesh and Ishak, though the graduates they are pursuing the respective colleges. Because of the internals, they are not able to join that. So moving to the things first. So nowadays, even the recent in Vijay, one of religious function, one person was died of the sudden heart attack. So I am the victim of the family where I lost my father, sudden heart attack, and I'm not able to differentiate whether it's a gastric acidity and the heart attack. So that's why that drives me to present this as an AI-driven heart attack prediction. Coming to the things was just to understand in a simple manner. So right now, earlier, we know we are getting the what I can say, news from the respective environment stating that we are getting rain so and so day or the temperature, whatever is going to happen. All these are based on the prediction of the existing data. They are using the existing data and the analyze and according to the conditions. So the similar concept, the similar approach here we are proposing that I will directly moving back to the main framework. This is an input layer of here. We can see the patient data, that's an ECG and EHR, biomarkers and lifestyles. So nowadays even the students, those who are studying or the working either any job market, because of the stress or the lifestyle, the type of food that we have consuming every day. All these are leading to the sudden heart attacks. That will be the input for this. And by pre-processing and nesting was just to be, it was an idea already developed just to be as a part of presentation I have taken, and by analyzing the existing model, whatever the research was happened, thought to take as an of idea to present for my future PhD work. So that's the reason by using the deep neural networks or the models that we have, there will be score was given to the risk factor. So based on the risk factor, we can say whether it will be happen in future, how much. There will be a few predictions will be done already by the Microsoft and the Apollo hospitals. So I don't want to quote the data just to be sharing the thing. So while consuming this, we need to take care of a couple of things with respect to the privacy of the data privacy, the cyber securities, and there will be some complex regulation that we need to take care of the HIPAA and respective regulatory agencies. So this was my idea for the prediction of the AI framework. Thanks for giving the opportunity to the management of my Infosys and the Andhra University in the Ravi sir. Thank you.
Has anybody got any questions? Thank you. Next up we have Navina.
A pleasant way. Good afternoon, sir, madam, and for participants. I am very thankful to give me this opportunity. Today, my project on sustainable agriculture mulching seeds from chitin-based bioplastics, sir. We all know how much plastic affects the world's present. To prevent the effect of the plastic on the agriculture, we introduced this project. Firstly, we know about how can plastic affects the agriculture. Agriculture uses the mulching seeds in the crop yields. That continuous usage of plastic in the agriculture can cause the releasing of microplastics. Microplastics are nothing but the molecules, small molecules which are released from the plastic. They can cause of the soil pollution and crop yield reduction and microplastic ingestion and the increase of the greenhouse emission. They all are impact on the biodiversity. To replace the plastic mulching seeds, we invented the idea bioplastic mulching seeds. Bioplastic mulching seeds is nothing but mulching seeds are prepared from the bioplastic. Bioplastic is plastic is prepared from the biological materials. The biological materials are chitosan and pectin. The combination of chitosan and pectin, we are produce the bioplastic mulch seeds, which can improve the soil health, increases the crop yield and useful for agriculture. No effect to reduce the plastic. The process involved in the preparation of the mulching seeds is explained by my friend Navina. Preparation of raw materials. First, we have collected the shrimp strays from the fishing harbour, Visakhapatnam. After that, we clean thoroughly to remove the excess debris. After that, we dry in sunlight for 1 hour. After that, the shrimp cells were dried in oven up to 60 to 80 degrees Celsius for 2 to 3 days. That we are in college. After that, we grind it into fine powder. This powder is raw material prepared by us. After that, we have done demineralization process. Demineralization process is the process to remove the inorganic materials present in it. We have added HCl, which was in 2 molarity. It was added in 1:10 ratio. We stir it 30 to 40 minutes under magnetic stirrer, and we also done the processing by using the sonicator. Sonicator takes less time compared to the magnetic stirrer.
Now I will share you how we prepare bioplastic by using the chitosan obtained from the shin cell. And we add water and vinegar in the 2:1 ratio, and we added plasticizer. Here we use plasticizer as a glycerin as a plasticizer. Currently, we have completed our project up to here. Our next step is to combine the pectin and chitosan to make a bioplastic. This combination of bioplastic enhances the mechanical strength and flexibility and providing the durability. After successful completion of pectin and chitosan based bioplastic, our aim is to prepare agricultural mud sheets and then check the parameters of bioplastic like tensile strength and bearing capacity, puncture resistance and thermal stability. And we have taken these journals as a references for our project. And I would like to thank my professors Karthik and Govind sir for guiding us every time during our project. And thanks to my project team and my friends, they helped us a lot. And I would like to express my sincere gratitude to the Vlse for giving us this opportunity.
Any questions?
So what is the strength of this bioplastic? I mean, can you make something, a stand like this? Does it have that kind of strength? Have you tested the strength?
Our project is currently under progress. We will complete this by the end of our master's program. We will work upon it. It was done up to the chitosan extraction.
What my question is that, is this self-sufficient or do you have to add some additives to make it more strong? What do you think? Do you think this is going to be strong enough by itself, or do you think you have to add more additives to make it strong? For example,
Sir, actually mulching seeds are the protective layers which is placed on the soil to protect the soil moisture and fertility. By using plastic, they can affect the plants. To prevent that plastic damage to plant, we replace it with the bioplastic. Due to bioplastic, no soil corrosion and good yield production and a good health, good can abstract from the plants. There is no need for standing like this. They are only placed on the soil like placed on that.
And based on the percentage of glycerin, we will make models like you said.
okay, so I guess you haven't tested that yet. Okay, thank you.
Combined to form more durable and strength.
Okay, so that is what I'm asking. That are you going to make it more strong,
Yes.
more durable?
For more strongness, we add pectin. We are going to combine chitosan and pectin.
Okay, thanks. Any more questions?
next two are we have Vaishnavi. Yeah, next up we have Darshan, and after that we have Miss Vaishnavi talking about Cetacos. And after that we have Divyashri. Okay, so please be ready.
A very pleasant good afternoon to all. Especially I extend my thanks and gratitude to the team for giving me this opportunity, and especially thank my lecturer Shabana Jasmine and my college my mentors for the guidance for this project. And this project, comprehensive evaluation of antibacterial and antifungal susceptibility testing. So this is my master's project, which is in progress. So basic. Our project overview is to study, as we all know, these days nowadays are microbes. So here I took Staphylococcus aureus and Aspergillus niger. So these microorganisms are becoming very resistant to the drugs we are using, like azithromycin, fluconazole or meconazole, drugs like this. So I got a thought that, so what could be the alternative for this? So how resistant are these bacteria and fungi are becoming to the drugs, and what are the alternatives we can choose instead of these? That's my project. For this testing, I've used susceptibility, antimicrobial susceptibility test. So what is the role of this antibacterial susceptibility test? First of all, it has a very crucial role in drug discovery. So as we all know, while we are designing a drug or we are discovering some new substance or new formulation against these microorganisms to prevent or to cure the microbial infection. So before that, we are supposed to test it, right? How resistant or how sensitive these microbes are becoming to this newly discovered drug. So this test will be used for the testing the newly discovered drug, and it has many more advantages, and it has a lot of background for this. And going to next one. In this project, I've used this diffusion, well diffusion, and blood diffusion methods to test the sensitivity and antimicrobial susceptibility. So basically, this diffusion is a very common thing we will be seeing, we'll be doing in our laboratory, so that. And for this studies, I've used. For these studies, I compared the synthetic drugs, pharmaceutical drugs of antibacterial, antifungal, and I compared this with the natural products like medicinal plant extracts. So for that, I've chosen penicillin, tetracycline, ofloxacin, and ciprofloxacin I've chosen these 4 antibacterial drugs. And coming to antifungal drugs, fluconazole, meconazole, echinazole, and ketoconazole, I've used these. For this, to test, these drugs were tested against Staphylococcus aureus in nutrient agarbinium and Aspergillus niger, which was grown on as per Zepectox agar. So before also, as synthetic drugs were already prepared, to compare this, I've chosen neem, goa, and holy basil and betel leaves to conduct this comparative study. So I've tried those leaves on a brief thing. I've collected these leaves, dried, powdered them, and I've incubated these powders, 2 grams of this powder with 10 ml of methanol, and used watermelon filter paper pieces to as discs for the further studies. So basically, I've used this diffusion method. So I've prepared agar plates, incubated, used spread rate technique for to study the antimicrobial susceptibility, and then place these discs, antifungal tablets and antibacterial tablets along with this medicinal plant extract discs. I've placed these discs in the center of the agar plate and see the susceptibility testing. Same with antifungal and antibacterial. So among these things, in synthetic drugs, penicillin have shown a good and well 19 mm of zone of inhibition on agar plate. And coming to antifungal, meconazole has shown 13 mm of zone of inhibition, and the rest of the drugs were being effective on that, but not as much as these drugs. And now coming to the medicinal plant extracts, in antibacterial, betel leaf has shown 17 mm of antibacterial activity, and later there are neem, holy basil, and betel leaves have shown different zone of inhibitions. And coming to antifungal properties are goa plant. Goa plant has goa leaves have shown 14 mm of zone of inhibition, and rest of the plants like neem, holy basil, and betel leaf, which is a big shock to me. The main reason I've chosen these 4 plants as neem, holy basil, which is tulasi, we all know, and betel leaf, pamalapaku. So these, we'll be hearing that these they have very well antimicrobial activity. So from our ancestral times, we have been using these 3A leaves or these 3 plant extracts for different uses like skin rashes or antifungal as an antifungal and antibacterial diseases, infections. But there's a big shock for me. Goa has shown, and Goa has 14 mm of zone of inhibition, while neem, holy basil, and betel haven't shown any zone of inhibition. They haven't shown any sensitivity to this Aspergillus niger. That was a big shock. That was the main thing. That's the main aim of my project. What alternate I can use for instead of synthetic drugs. And in summary, I want to conclude with this. As penicillin, meconazole have shown very well and good amount of sensitivity against these microorganisms. Yet these medicines, these microorganisms are becoming resistant to this. Day by day, they are. If we are increasing the dose of the tablets or increasing the dose of these medications, they may show any side effects. So, comparing notable plant extracts, like betel leaf have shown a good antibacterial activity, and goa has shown very significant antibacterial as well as antifungal activity on antifungal and antibacterial activity against Staphylococcus aureus and Aspergillus niger. So on a whole, I want to conclude this. Synthetic drugs are well, they are good for treating these infections against antibacterial or antifungal infections. But our natural products like goa, it have both the effect, both antibacterial and antifungal effects. So using Goa instead of Goa leaf extract in therapeutic drugs could have a significant change in significant change in therapeutic drugs history or future. So, from this study, I've seen there are many. I've searched go through the web for. Are there any drugs or therapeutic drugs for Goa-based, plant-based therapeutic drugs? I haven't found anything. No Goa plant-based drugs are present in the market. So this study can help. This study can conclude that combining natural products with synthetic or in pharmaceutical drugs can show a good effect on antibacterial and antifungal activity. So my further study takes me, so as science is all about trial and error method, so my next further study will be on combining this synthetic drugs and natural product, and that should be done in my further studies. Thank you. Thanks.
So you mentioned extracts. How did you extract them?
So, first of all, I have collected the plant leaves. So these studies are in this study for till now I have used only leaves. I still have to use different parts of plant, but till now I've collected the leaves and I've air dried them, not sunburn. I haven't dried them under the sun. I've just air dried them in a hot air oven and powdered them. Now what I've done is took 2 grams of plant leaf powder and incubated it with 10 ml of methanol overnight, and then I would and then I've used Wattman filter paper 1 and made some using this paper and soak these papers in this this solution of plant extract solution, and I've used those papers as instead of antibacterial discs.
And when you actually did this, have you used any other solvents or just methanol?
I've just used methanol.
So is methanol hydrophilic or hydrophobic?
Methanol is hydrophobic.
Okay. And did you use any hydrophilic solvents?
No, I haven't used any hydrophilic solutions.
Okay. And we don't have any quantification here. We don't know how much of your guava, holy basil, which is also known as tulsi.
Yes, sir.
And neem. You don't have any concentration dependent measurements here.
Yes, I haven't mentioned it.
And it's the same problem with the control standards. You mentioned about the ciprofloxacin, ketamazole, all these things. What is the concentration of each of those tablets? Because the zone of inhibition depends on the concentration of the antibiotic. So if you add more antibiotic, you will get a bigger zone of inhibition.
Yes.
So without mentioning the concentration, it's hard to estimate things.
So for that, I've taken every tablet of 10 mg of tablets, I've crushed them, and I've combined with ethanol and used same process as a plant extract. I've used a Wattman filter paper, I soaked them in it, and I've done the same procedure.
And what makes you think that you got the pure active ingredient into that Wattman filter paper? Because the tablet contains excipients and the drug.
Yes.
So it could be excipients also getting into the filter paper.
My study wasn't yet concluded, so I'll take your suggestions into consideration and follow.
My point is, it's good that you've done quite a bit of work, but it has to have certain aspects that you have to quantify the amount of the drug you are using, and then you have to clearly mention that you are using 10 mg or getting so and so millimeters of the zone of inhibition. Yes, that kind of quantification, if your control is not quantified properly, then you cannot talk about your leaf extracts.
Yes.
Right? Yes. Yeah, and methanol is actually hydrophilic, by the way. Any more questions? Okay.
therapeutic small molecules against Chlamydia psittaci OMDA. Recently, we got, sorry.
So anyone here, please don't feel stressful. Nobody is going to kill you. This is supposed to be, like a fun event where you are talking about science and people are here to enjoy the science, finding the story that you are going to tell. You can also explain it in Telugu if you have difficulty in speaking in English. Okay, please don't choke yourself. Relax, chill.
So next up we have Divishri.
Good afternoon everyone, this is Divya and I am from CSA Chepalpur, Andhra University. She is Tanuja, she is from CSA Chepalpur, Andhra University. So today we are here to present our AI model. So imagine two scenarios. Like you're scrolling through Instagram and you find out a video of a well-known celebrity saying something shocking and it's completely fake. And imagine second scenario, which is you're receiving a text from your friend like asking for help or they are requesting you to send money as your her friend and something. And you think that it's real and you send money and you lost it because it's completely fake, as she has created an account which she uses a deep fake image to trick you to get money. So this is our main motto. We have created this idea. So to tackle these types of problems, we have proposed a solution which is AI model, which is true lens deep fake scam detection, where we trained thousands of images which are real and fake. And using Resident 50, which is the CNN layer, we have proposed this AI model, which is where we upload an image and it detects whether the image is fake or not. And these are why deepfake detection. So rising threat for misinformation, like the celebrities which we see, like Rashmika Mandanna and all, they are being mocked, like the body is different and the face is different, they're attaching, and some influencers are getting more followers because they're getting popular by seeing these images. And real world relevance. It's very difficult to identify which is real and which is fake. Application of AI. So this is the model where we have created using AI, and AI can be used in both positive and negatives. Like we have created this solution based on AI. Based on AI, they have created the deepfake images. So I think many of you have used Deeply Arts, and you can see ChatGPT and Perplexity, Gemini creating images which are completely fake. Like you are giving your personal information to that, and that is generating images which are completely fake and ethical and social impacts. So this problem is not just a technical problem, it's completely about personal life, a person's personal life which is being disturbed by getting a single image which is morphed. And scope for innovation. So we are planning to include, this AI model is completely based on images. So we are trying to make this model work for even the videos and real-time messaging. Like in video calls, it detects whether it's fake or not. And personal motivation. So I think a single image can decide a person's life, whether it's fake or real. So the tech stack we have used is Python as the main programming language. We have used entire this to make this to build this AI model. And the frameworks like TensorFlow and Keras we have used. We have used ResNet 50, which is the TensorFlow model, where we have imported it from Keras. And Streamline is used for building the interface, website interface. And NumPy is used for processing the image arrays, and the Pillow, which is used for image data processing and image processing. We have executed this program in VS Code.
So coming to the workflow of the model, first we'll be giving the input image, and it will pre-process the image. That means it will cut the image into 224 frames by 224 frames, and then the feature extraction. That means it visualizes the image with the features of the image. Like if for a normal picture, the features will be different, like near a face. It's not like a real image, no? So it will extract the feature and it will compare with the pre-trained model that which we have given, and then it will visualize the output, and it will show that if the output, if the image is real or fake. Challenges faced during this project. Actually, when we were doing this project, we didn't get that accuracy at the starting stage. We got till 60 to 70% accuracy, but as we gave so many pictures and we trained the model, then we got the accuracy, and the accuracy is at 96 to 97%. And due to the subtle differences in the images, comparatively to the real and fake images, nowadays we are not, they're not that clear. We can believe that a morphed image is a real image. So due to that subtle differences, the accuracy will be lost. For that, we need to train the model in a better way. And to collaborate the code and all, there was some difficulty. That's it. Uses of our model. Social media verification means in social media, so many people will mark the images in the thumbnail for getting views and all, so that we can get a verification that it is not real. And news and media authentication, if the news is real or not, and public awareness and education that we should not believe in such morphed things. Case studies. In real life, we saw that Rashmika Mandana's deepfake photo and Katrina Kaif, but recently some photos, like morphed photos. As their celebrities, they have that like privilege to not face any problem, but a common person will cannot face that situation and he will go against the life. So for that only our model is helps. And if we see recently that Rashmika's morphed pic, and because of this deepfake, they made Narendra Modi also dance, and so many thumbnails also are morphed. So impacts and benefits of this project is we can prevent fraud and cyber security enhancements. Means it strengthens the digital safety by detecting if the image is real or fake, and we can build a trust and ethical AI promotion we can use AI in an ethical way. Future implementations. Till now, our model only takes photos of any size, but in future we can make our model even take videos, and it also detect audio and text and video in future, and real-time fake detection in live video calls. In live also we can detect if the other person is real or fake, and we can extend this to as a mobile app also. And blockchain-based media authenticity for verification, integration with law enforcement, cybersecurity systems, means we can integrate with cybersecurity and we can make it even more secure. Social media API integration for automatic content scanning, means to connect with some API integration, and we can directly scan if the content is fake or real. Thank you for giving this opportunity, and if there are any doubts, you can ask this.
Any questions? So how many detections have you done using your model so far?
We have a demo video.
Next up we have Varshini. And the next two participants are Yusuf and Pujita. Good evening to respected dignitaries, faculty members, and my dear fellow students. We are from Dr. V.S. Krishna Government Degree College, and myself, Devavarshini, here my teammates Malvi and Uday Kiran. Today we are excited to share great idea, great area. Today we are so excited to share a great innovation idea about bioluminescent algae by using sustainable city lights.
What is bioluminescence? Bioluminescence is the natural ability of living organisms to produce light through chemical reactions. Here, chemical enzyme refers to luciferase, which acts on the molecule luciferin and emits light without any heat. We can see this in fireflies and also some microorganisms such as Noctiluca and Pyrocystis fusiformis, a glowing algae of the sea. So the idea, urban lighting from living algae. So imagine instead of having electric street lamps are being replaced with bioluminescent algae panels or water channels that can emit light at night time. So these microalgae can be cultured in transparent tubes or biocanals that can be placed in parkways or inside the parks. When they are touched or moved or disturbed in the night time, they emit soft blue-green glow, a living, breathing light source.
How it works? It's a very simple method actually. It's a very basic setup. It contains a bioreactor tube or a tube system which contains very high rich medium along with the algae. It's a very important for the algae to grow as early as possible. And as we know that during daytimes, algae uses carbon dioxide and emits an excellent oxygen. Lots of oxygen we get from algae. And during night times, when they get disturbed, it will emit a bluish light by their naturality. By their nature, by its nature, it releases a blue light. It's a very excellent light. It's very visible for everybody. And also we have real example in bioluminescence lights, which was in France, I think. They use that bioluminescence lamps in streets and also in parks for the glowing benches. When it comes to the advantages, it's a very excellent eco-friendly method actually. That means no more carbon dioxide emission and no more electricity. And it's a very beautiful method actually to use in public parks, which will add some more beautification for the parks and especially for the coastal cities like Visakhapatnam. Currently, VMRDA is going to develop the Visakhapatnam. This is also a good idea to improve, to make the city, to add some more beautification for the city. And it's an excellent air purifier actually. It absorbs a lot of oxygen and gives, it absorbs a lot of carbon dioxide and gives oxygen to everyone. And also when it comes to the educational institutions, it promotes public awareness about the biotechnology of marine and gives the renewable designs. And also when it comes to the challenges, actually they are very less lifespan species. And we have to culture them in a restricted areas like temperature, certain low bright. They provide very low brightness actually, and we can culture them in a very low pH. And it's somewhat of cost needed thing actually. This is all about the bioluminescence by the nature species especially. And we are so thankful for being here and we are feeling so great to get this opportunity. Thank you so much sir for giving and providing and encouraging such youngsters for the new innovations. Actually we came to know this
very in a less time because of having semester examinations. We are more concentrated to the. So this is all about, because of having less time, we are concluding our topic in a less manner. So this is all about the bioluminescence by the nature. Thank you very much for everyone for your great patience.
Any questions, anyone?
Bioluminescence की ఒక type of light అంత bright light అయితే ఉండదు. what means దానికి దీనికి మీరు కాంపిటేట్ ఎలా చేస్తారు? మీన్స్ నార్మల్ లైట్ దీనికి you want to replace that.
So we are not saying it's a perfect replacement, but we can use it as a supplement. Especially in tourism places ఉంటాయి కదా, beaches కానీ parks కానీ చదువుకునేంత లైట్ ఇవ్వకపోవచ్చు,
but in the night time it's difficult కదా.
కల్చర్ చేస్తే అవుతుంది. నెక్స్ట్ ఆర్డియన్ టెక్నాలజీ యూజ్ చేస్తే మనకి
But
కొంచెం
how much time will it give the light?
not much time, but
In a very less time we can get the light. When they are distracted or disturbance, we can get that light. But here, remaining time we don't get. For the adding of beautification purpose, we are only taking this concept, not for educational, institutions or companies. You can't read even under the light of God.
Okay, I guess the discussion is going in a different direction. So it is clear that it is not as bright as the regular lights. And also it requires the agitation. You have to disturb it, right?
Yes.
Those problems can be solved biologically. But temporarily, a solution that you can use is you can just use a small piece of that algal tube, whatever, and then you put a bunch of mirrors. So those mirrors will actually reflect the light and it actually intensifies it. It improves the brightness of the light temporarily.
Yes.
Any more questions? Okay, thank you.
Thank you so much for everyone.
So we are going to look at the demo video of the previous presentation.
So this is how our AI model works. So first we upload an image which is in format of JPG or PNG. And first we have taken real image. So the model analyzes it and it shows real. And it shows the confidence score, that is 70.48. And next we have taken another real image. The model analyzes it and it shows real of 90%. The next two are fake images which are generated by AI. Which is deep fake of 32%. And next it is AI generated image which shows deep fake.
So how do you guarantee that your initial image was not fake? How deep did you train your model? let's say I take a picture of one of you. No, listen to me. I take a picture of you in the darkness, right here, and in the bright light. Is your model capable of detecting whether this one is fake compared to that one?
If the image is clarity and the background is good, then it shows to be perfect rate, perfect percentage, it shows really. If the image is it's good, but pixel is clarity.
Okay, so you are based on pixels, but pixels can be manipulated.
Based on what we have done, what pixels have put on the paper, by comparing with it, show that how much percent fake or fake.
That's what I'm saying. How do you know whether the original image that you're training your system is original? What if you train your system with the fake image?
Then it shows fake, because it doesn't have less than we give something.
That's what I'm saying. So what have you done to tell your system what is the real and what is the fake? Like how deep did you train your system?
To be 2 lakh images we have trained. Actually, there is a graded model which contains 50 layers, 50 neural networks. And we have added extra 5 to 6 layers, which is dense and dropout layers with optimizers called as added. And we have a learning rate of 1 minus 4. So we have used those and we have trained with the 50 layers of dense layers.
Any more questions?
Thank you.
Next up we have Varshini. Is there a Varshini? Oh yeah. User Pujita. Good afternoon everyone, myself Pujita, this is Prasanna. We came up with the idea of a portable diagnostic kit. Multiplex lateral flow assay kit. This is portable kit that is lightweighted and can diagnose 4 different types of diseases: malaria, dengue and bacterial infections and anemia. This kit is capable of detecting 4 diseases at the same time simultaneously within a single kit. And the duration of detection is also short. This is cost effective, and here majorly we use 3 main types of samples: blood, urine and saliva. The major sample we take here is blood. The amount of sample to be taken is 10 to 20 ml of blood and 50 to 100 ml of urine or saliva. Now Prasanna gives a brief about the major components of the kit.
Major components of the kit and their importance. Gold nanoparticles act as visual reporters and exhibit a characteristic red color. We take a 30 nanometer gold nanoparticles to detect red color. Nitrocellulose membrane. It is a protein immobilization. It uses for protein immobilization. Conjugate flat stores bright genes with conjugated antibodies. Sample pad filters unwanted materials from the sample. Observation pad pulls samples through membrane via capillary action. Test lens, solution of monoclonal antibodies specific to target antigen. Control lens specify secondary antibodies are used. As test lens reagents, for malaria we use anti-PL-DH antibody solution. For dengue, anti-MS1 antibody solution, and for bacterial infections we use CRP proteins. For anemia, TMP solution. Test and positioning. Malaria, test and positioning placed at 7 mm from the sample bed. Dengue, 12 mm, anemia 17 mm, and bacterial infections 27 mm. All components must be assembled step by step. Remind me,
the major reasons for selecting particular type of antibody solution is for malaria we take PFHR P2 protein antibody solution because this helps in detecting antigens related to falciparum species of Plasmodium which causes deadly malaria. The important thing that we use here is a PLDH enzyme is released by all types of Plasmodium species which helps in detection of early infection. Anemia. In anemia we use TMP solution which detects the hemoglobin concentration in the blood sample. Bacterial infections. In bacterial infections we use CRP proteins. These proteins are generally released by our bodies. When any bacterial infection occurs, it helps in detecting within 6 to 8 hours of infection. It helps in differentiating between a bacterial infection and viral infection. For dengue, we use NS1 antibody solution because there are mainly 4 types of dengue viruses. All 4 types of dengue viruses release this NS1 protein and helps in detection in early stage. The mechanism involved in the test kit is we place the sample in the sample pad, then it gets absorbed where it gets filtered from the large particles such as RBC and large proteins. The sample gets migrated to the conjugate pad by the capillary action. The absorbed sample rehydrates the dried gold antibodies and gets mixed with the sample. These gold antibodies are useful for the color detection, whether it is positive or negative. The sample along with the conjugate mixture flow through the nitrocellulose member. If the targeted antigen is present in the sample, then it gets binded to the gold antibodies and forms a complex. It takes generally 20 to 10 minutes of time for complete migration and result. Test result determination. If the test is positive, that means if the blood contains antigens specific to that disease, then they get bind with the antibody solution that we used in the test kit and exhibit a prominent red or pink colored lines along with the blue or purple color control lines. If the test is negative, only blue or purple color control lines is appeared. No test line is appeared. If a test line and control line both are absent, then
your
the
time
test
is up. Yes.
Generally, this kit is lightweighted and pocket sized and can detect multiple diseases. No special equipment is required to detect this. This is as simple as a pregnancy kit and easy to use and long durability and cost effective. Thank you.
Now Sadhya will present her presentation.
going to talk about one of the ongoing works that are happening in our lab. Myself, Sadhya, and I'm actually an intern at the PCAP-ZE. So we are actually doing a project on testing the genetic similarity claims on HIV and COVID. So it all started during the early pandemic when actually a rumor came up that the SARS-CoV genome was actually artificially engineered from the HIV genome, because the SARS-CoV genome actually contains some HIV-like insertions. Well, as such was the claim. But most of the scientific community was clearly against this, or they clearly tried to debunk this rumor. And here in our lab, we try to, aim to test this claim directly using genomic DNA samples and HIV-specific primers. So this is the preprint behind the rumor, and it was taken down immediately. They claimed that the SARS-CoV-2 genome had 4 insertions in its spike protein, which are similar to the HIV-1 proteins. And they suggested these similarities are because the SARS-CoV-2 genome was actually artificially engineered from HIV. So the significance is we want to clarify misinformation, if any, with regard to the origin of the SARS-CoV-2 genome, and we demonstrate the use of PCR and sequencing in validating those genetic similarity claims, and we want to provide a hands-on understanding of the experimental validation of genetic viral relationships. So the hypothesis and the objective include that if the COVID genome actually contains any HIV-like regions, then the HIV's primer should be able to amplify those regions when performing the polymerase chain reaction. The objective is that to test the presence of HIV-related sequence in the SARS-CoV-2 genome, in the human genomic DNA samples by PCR using HIV-specific primers. So the workflow, the gist of the workflow that we have done is we have taken collected blood samples from the members of our lab itself, and then we extracted and isolated the DNA. Then we did a confirmation analysis using PCR to check if the DNA was actually isolated or not. And then we performed another round of PCR using HIV-specific primers this time. And then we performed agarose electrophoresis to check the bands, or if there was any amplification of those regions in the sample using the HIV-specific primers. And then the next step would be ideally to send if any positive bands for sequencing purposes. And we have actually currently sent those bands to sequencing, and we are waiting for the results. So methodology, we use the extraction of human the genomic DNA. We have taken them, we have extracted them, we have purified them, and then, like I said, we did PCR, and once the confirmation analysis was done, we did 11 times PCR of 4 blood samples that we finally isolated, so such that we had 44 samples with us. And this time we performed PCR at a wide range of annealing temperatures, starting from 50 degrees Celsius to 60 degrees Celsius using HIV-specific primers. Then we performed the gel electrophoresis across 3 gels, and we isolated those isolated product samples were then loaded into the gels for bands detection, and then the next step was to sequence if any positive bands were observed. So the results, this gel is actually the gel which was done after the first PCR for confirmation analysis whether the genomic DNA was isolated or no. The gel is split into 2. You have the upper part and the lower part, and the first columns in both the cases are the ladder DNA, and the remaining adjacent wells are loaded with our samples. And you can see bright bands in there which indicate that the DNA was intact and as of high concentration. And you cannot, we don't even observe any RNA contamination because there's no bands at the bottom part of the well. Then these are the results which we got after doing the PCR in the second round for 11 times using the 4 samples at different annealing temperatures. So these are 3 gels. We distributed 44 samples for obtained from 11 times PCR, and we put them into 6 parts of the gel, that is 2 in each gel. And like I said earlier, the first the first column is always the ladder, and the corresponding ones are the samples that we have loaded. So from that we could observe that the PCR in several lanes some very bright bands were observed. Such temperatures where these were observed were of 50 degrees Celsius, 54 and 58 degrees Celsius annealing temperatures. So the corresponding PCR products have been now sent for sequencing, and we are waiting for the results to actually verify the identity of those sequences. So this is like a summary analysis of the bands. Those are 4 samples across a wide range of temperatures from 50 to 60 degrees. And as you can see, 3 samples at 50 degrees Celsius bands were detected. The same thing, sample 1 was detected at 54 degrees and sample 2 was detected at 57 and 58 degrees Celsius. There were no other bands that were detected on other different temperature ranges. So conclusion was that we have successfully performed the PCR amplification and we have confirmed it with Gallows gel. But as of now, by our initial observation, no such specific HIV size PCR products were detected, and there were no clear or consistent HIV bands. So the sequence, whatever bands we have obtained, we have sent them for sequencing, and we are actually waiting for the identity of those bands. Thank you.
Any questions? Okay. So with this, we conclude the oral sections, unless you are ready. You want to give another try?
Please allow them to present in their language.
You can present in Telugu, that's fine. You know Telugu, right?
Please do.
Telugu మాట్లాడవచ్చా మీకు? తెలుగులో చెప్పండి. It's okay. See, one of the main reasons of Vlse Symposium is to make sure that people get a chance to go up on the stage and talk, so that you get rid of your fear. A lot of people have stage fear. This is the time that you get rid of your stage fear. This is like an icebreaker event for you. Okay. No? Alright. So we are going to move on to the poster sessions. There is tea available, and I just heard that the guys who were supposed to set up the mocktails this morning, they just came. So I guess they are serving mocktails also. So it's up to you. And also, I think we forgot to announce our internship programs. If anyone is interested in our internship programs, please come by and talk to us. You can send us an email, or you can talk to any of my students that are here. We have internship programs in life sciences, computational biology, and medicinal chemistry. And all these internship programs have 95% lab skills and 5% theoretical skills. So we don't lecture, we only do lab. It's all hands-on, which means the first day you come to the lab, you'll be given an experiment and you have to do the experiment. So that is how you learn the practical skills, because without practical skills there is no future. Theoretical skills are no more good. It's good to have knowledge, but theory cannot get you through a job. The job market is very competitive. As you can see, it's not just life sciences. It's the AI, the combination of life sciences. If you are not ready for life sciences, forget about the future, because the future is not just life sciences, because it is AI empowered life sciences. Okay? Don't feel the stress. All you have to do is start today at least. If you missed it yesterday, that's fine. Start it today. Make sure you know the latest, what is going on, what is AI. Not everybody understands what is AI. That's okay, it's not a problem. But you must know how AI is applied in life sciences, applications of AI. Because that is the future, that is the tomorrow. If you miss it out, you are going to miss out the next 20 years, the whole generation. So then you'll have very difficult situation for your career. So make sure you start thinking from today. I hope you got good experience today with different talks. You've listened. I actually wanted to give a talk about our achievements, but I kept it short from a small video. I hope you guys saw the video. So my talk will definitely take more than 1 hour, so that is why I did not involve, because Vlse is to give you a chance to come upstairs, to come onto the dais, break the ice, start talking, get rid of your fear. Okay? Alright, so let's move on to the poster session. Thank you all, whoever did the presentations. And after the poster session, we will do the prize distribution, and that will be the end of today. Thank you.
నువ్వు భయపడి వచ్చింది వెనక్కి వచ్చేయడం వచ్చింది. చేసుకోవచ్చు కట్ చేసి ఎండ్ చేసుకోవచ్చు. శవానికి వేస్తారు.
రేపు మధ్యాహ్నం వాళ్ళకి ఏదో వేరే ఈవెంట్ ఉందంట సార్ తెలుసా?
ఎవరికి?
వీళ్ళకి ఈ హక్కు వాళ్ళకి ఏదో ఈవెంట్ ఉందంట. కోసం మాట్లాడదామా? కృష్ణ పంచాయతీ.
Thank you.